Welcome to the development of Drosophila module. In this talk, we're going to learn about the different genes involved in the development of Drosophila. Drosophila is a protostome that belongs to the phylum Arthropoda. All insects and arachnids come under this phylum. In the case of Drosophila, the egg and the sperm fuse to form a zygote, and that zygote then undergoes the process of cleavage to form a multinucleated embryo. The cleavage is superficial cleavage, and hence it is a type of meroblastic cleavage. Once the multinucleated embryo is formed, a process of cellularization occurs to form the blastula. During the process of gastrulation, cell movements will occur, and we end up with a segmented embryo that will hatch from the egg to form a larva. The larva then forms a pupa, which can then undergo metamorphosis to form the adult fly. There are different groups of genes that control embryogenesis. The first group of genes are the maternal effect genes, and these are the ones that are expressed first in the developing organism. Thus, early in development, when we have the syncytial blastoderm, the maternal effect genes are expressed and they tend to form gradients. The maternal effect genes then aid in the regulation of zygotic segmentation genes. The first zygotic segmentation gene that is expressed is called as the gap genes. The gap genes then aid in the expression of the next class of genes, which are the pair rule genes. The pair rule genes are the genes that are going to cause or aid in segmentation of the embryo. The pair rule genes then aid in the expression of segment polarity genes. Once the segment polarity genes are expressed and segmentation occurs, homeotic selector genes are expressed, which aid in the formation of different structures of the organism. The maternal effect genes are deposited in oocytes in the form of messenger RNA during the process of oogenesis. The messenger RNA are translated to form proteins, and these proteins form gradients in the developing organism. The maternal effect genes play important roles in the formation of the anterior posterior as well as dorsal ventral axis during development. Maternal effect genes also regulate the expression of the first set of zygotic genes that are expressed, which are the gap genes. Let us now look at the first set of the segmentation genes, which are the gap genes. These are expressed in one or two broadband domains along the anterior-posterior axis. So if you look at the figure that's there in the blue box, we can see the different expression patterns of the gap genes. They are noted in color and their expression bands are shown in the appropriate color. For example, tailless, which is shown in purple, we can see its expression in purple, whereas giant's expression is shown in green. So as we can see that their expression is formed in the form of these broad bands, and you can have either like one broad band like that of Kruppel, or you could have two broad bands like that of NERPs or tailless. Now, when it comes to the initial gap gene expression, it is actually mediated by the maternal effect genes, but their expression is stabilized and maintained by repressive interactions between different gap gene products. Now, if we have a mutation in any of the gap genes, we actually see gaps in the development. And that is why these class of genes were called as gap genes. So for example, if we look at the figure to the right in the blue box, let us say that one of the gap genes gets mutated. Then in the blue box to the right, we are seeing the normal segmentation pattern of the embryo. And if that specific gap gene, which is Kruppel in this case, undergoes mutation and is lost, then we end up with an embryo that basically has lost the orange portion or the peach portion of the larva, and thus there is now a gap in its development. The same can be seen when the gap gene NERPS is mutated. Now in this case, the embryo has lost other parts of its body and hence there is once again a gap. The gap genes aid in the expression of the next segmentation genes, which are the pair rule genes. The pair rule genes are expressed during nuclear division cycle 13. What should be remembered is that the expression of the pair rule genes indicates the beginning of the segmentation process. So in the case of Drosophila, the moment the expression of pair rule genes occur, that is when segmentation starts happening in the embryo. The different concentrations of gap proteins can determine whether the pair rule gene will be expressed or not in specific locations of the embryo. The transcription patterns of these pair rule genes divide the embryo into regions that are precursors of a segmental body plan. Normally, they are expressed in alternating parasegments. So if we look at the figure that's there in the blue box, we are looking at the expression of two pair rule genes. The one in red is called as even skipped, and the other one that is in purple or gray is called Fushitarazu. When we look at the expression of even skipped, we can see 7 stripes. When we look at the expression of Fushitarazu, we also see 7 stripes. However, the 7 stripes of Fushitarazu do not overlap with the 7 stripes of even skipped. And thus, we will actually see 7 stripes that are the expression patterns of pair rule genes. And in many cases, we could have alternating patterns. There are 2 types of pair rule genes, which are the primary pair rule genes and the secondary pair rule genes. The primary pair rule genes, their expression is mostly regulated by the maternal and gap genes. However, in the case of the secondary pair rule genes, their expression is normally regulated by the primary pair rule genes, and thus the secondary pair rule genes are normally expressed afterwards. Now, when we look at the genes of the pair rule genes, they have multiple enhancers. And there is a general rule that states that there will be an enhancer for every stripe that we see in the pair rule gene. So in the figure that is shown below, we are seeing the gene for one of the pair rule genes, which is even striped. And in the blue in the middle is the coding region. And the other color-coded regions are the enhancers. Now let us say, for example, the first enhancer. Now this enhancer is responsible for the expression of the pair rule gene in the stripe number 2 and stripe number 7. When we look at the next enhancer, it is responsible for the expression of the pair rule gene in stripe number 3. In the case of the one that's in the blue box right now, that enhancer will control the expression in stripe number 4 and 6, while the last one that we're seeing, we can see two stripes. One is for stripe number 1 and the other is for stripe number 5. And thus these multiple enhancers are able to regulate the expression of the pair rule genes in their appropriate stripes. Now, how did we actually find that out? So what was done is the enhancers were taken, so the DNA sequences that correspond to the enhancers are taken, and the example here that we're looking at is that of 2 such enhancers. One is for stripe number 1 and the other is for stripe number 5. And now what we're going to see is we will take, let's say, the enhancer for stripe number 1 and put it under the control of a lag Z. Now, when the lag Z gets expressed, if that enhancer gets activated, then we're trying to see where can we observe the expression of lag Z. And in this case, we can see in the blue box that it is the first stripe that looks really, really dark. And thus, this is an enhancer that regulates the expression of the pair rule gene at the first stripe. On the other hand, if we take the enhancer for stripe number 5 or the DNA sequence for the enhancer for stripe number 5 and join it to the lag Z gene, then when we look at the expression in the embryo,
we can see the fifth stripe is where we observe the lag Z expression, and hence that is where the parallel gene is expressed. The same can be seen if we put the whole enhancer, which corresponds to stripe number 1 and stripe number 5, that have been the DNA sequences are joined to lag Z, and in the resulting embryo we can see stripe number 1 and stripe number 5 are very dark, and that is where lag Z is expressed. So this is the way people actually found out different enhancer sequences and where they were. The parallel gene products activate the segment polarity genes. So whatever proteins are made by the parallel genes, they are the ones that will activate the expression of the next set of genes, which are the segment polarity genes. The segment polarity genes are expressed after cellularization occurs. They reinforce the parasegmental periodicity and establish the boundaries, like the anterior and the posterior boundary of the parasegment. The segment polarity genes encode proteins like Wnt and hedgehog and the proteins that are involved in their signaling pathways. In the case of Drosophila, Wnt is actually called as wingless. And so if you're going to be reading some papers or information about development in Drosophila and you see wingless, it's basically the Wnt protein. The segment polarity genes aid in setting the anterior and posterior boundaries of the parasegments, as I just mentioned a few minutes ago, and they also aid in the development of the final segments during embryonic development. Another set of genes that play a very important role in the development process are homeotic selector genes. Now, after the segmental boundaries are set, the gap and parallel genes regulate the expression of a class of genes which are the homeotic genes. The homeotic genes specify the characteristic structures of each segment, and thus they are responsible for specification of the fly body parts. In other words, what kind of body part is determined by which segment is what the homeotic selector gene does. Now, most homeotic genes are found in two regions of chromosome number 3, and hence that region which has both parts of the homeotic selector genes is called as the homeotic complex, or HOMC. We actually see two regions, and the first region is called as the antennapedia complex, and that is the part that is shown in the blue box. It is the complex that specifies the head and thoracic segments in the developing embryo. The second region of the chromosome has the bithorax complex of genes. This region will specify the third thoracic as well as abdominal segments. Now, mutations in homeotic selector genes results in changes in phenotypes, and in many cases, the phenotypes that we observe are honestly quite bizarre. So let us look at what happens if there is a mutation in one of the genes that is called ultrabithorax. In this case, when we look at the picture, the fly to the left is the normal wild-type fly, but the fly to the right has a mutation in ultrabithorax, and it is shown inside the blue box. In this case, what is happening is the third thoracic segment actually does not develop very well, and instead the second thoracic segment gets duplicated, and that is the segment that has wings. And so we end up with a fly that actually has 4 wings instead of 2 wings. Another mutation is what we can observe when we have antennapedia mutated. And in that case, what we see when we look at the picture to the left, we're looking at a normal fly that has antenna. When antennapedia gene is mutated, we actually end up with a fly that has legs coming out where antenna should be coming. And thus the homeotic genes help in determining different body parts of the fly from their appropriate segments. With this, we come to an end of all the different classes of genes that are involved in embryonic development.
With this, we come to the end of our talk, where we learned about the different groups of genes that play important roles in embryogenesis. We also learned about the regulation of these groups of genes and the expression patterns.